No. Hi, everyone, and thank you for taking the time to view my presentation. My name is Ryan Wirt. I am a interdisciplinary neuroscience student here at UNLV, and I'm really excited to share with you some preliminary data from my dissertation project, where we're examining the effects of alterations in blood glucose on brain activity. So I want to start with just a little bit of background about diabetes, which is a chronic health condition that affects how the body turns food into energy. In type 2 diabetes, the pancreas makes less insulin, and that can cause spikes and drops in blood glucose levels, and in some cases, mild cognitive impairment. It's an extremely prevalent disease, and predictions suggest that by the year 2030, more than 10% of the global population will have diabetes. Importantly, people with type 2 diabetes are more than twice as likely to develop Alzheimer's disease later in life. Alzheimer's disease, which is the most common form of dementia, is characterized by the presence of beta amyloid plaques and neurofibrillary tangles, which you can see in the animation on the right. And in some cases, very severe cognitive decline and cognitive impairment, with symptoms such as memory loss, inability to make decisions, and impaired spatial navigation. So the aim of this project is to try and understand what's going on in a diabetic brain that is leading to the development of Alzheimer's pathology. To this end, we utilized a rodent model of diabetes that was implanted with a 64-channel in vivo recording device that our lab designed and built to target the hippocampus and frontal cortex of the rodent brain. And so after we affixed these implants to the animal, they had sufficient time to recover. They were placed into either an experimental group, which received streptozotocin injections, which is a chemical known to impair pancreatic beta cells, which are the insulin-producing cells in the body, or a matched control group. After the subjects exhibited prolonged hyperglycemia, we attempted to treat that hyperglycemia or elevated blood glucose with a compound known as Florazin, which has previously been shown to reduce blood glucose levels. To measure spatial learning and cognitive impairment, subjects were trained on a conditioned place preference task, which, simply put, there's a neutral environment with two separate sides. We expose the animal to the environment, make them familiar with it, and then we bait one of the sides with a chocolate reward and train them on that side, and then the other side has no reward. And it is expected that if the animal was able to learn the environment, they would spend more time on the rewarded side. So the first thing we wanted to look at after we did all of these procedures was how our STZ and PZ treatments worked on blood glucose. So in the top graph here, you can see blood glucose levels measured over time in our STZ or vehicle group. The cyan is the STZ, and the smaller, thinner lines are actually our individual data points for all the subjects in this experiment. What you can see is there's a dramatic increase in blood glucose levels in our STZ group, while the vehicle group or control group stayed relatively stable throughout the experiment. Around day 79, we actually started using Florazin or PZ on our subjects, and you can see there was a mild reduction in blood glucose levels, but we did not find any significant changes with our current treatment, but we are planning to adjust the dosage and try and get that a little bit better with our follow-up experiments. Below, just looking at subject weight over time. Again, the larger, thicker lines are the group means, and the smaller lines are individual data points. And all we're showing here is that both STZ and control groups did stay relatively stable weights, so we didn't have any very sick animals or anything throughout the experiment, which could actually alter behavior or electrophysiological data in our follow-up experiments. So looking at our behavior, we found that both STZ and Florazin reduced spatial learning. However, STZ group was only trending towards that towards significance, while both the fluorescein vehicle or STZ group did exhibit significant spatial learning compared to our control animals. The spatial learning score is just, we created it by taking the amount of time the animals spent during their first exposure in the environment before training and subtracting that from the amount of time they spent on that side of the environment post-training. On the right, we're just looking at running speed in arbitrary or pixels per second. This is just to show you that there was no significant changes in overall behavior. All the animals were moving at the relatively same speed, and it's not like an STZ or fluorescein animal was just staying still the entire time. So after behavior, we started looking at what kind of changes we observed within the brain of the hyperglycemic animals and the fluorescein-treated animals. And what we found is that the oscillations or the rhythm of the hippocampus seem to increase by a little bit. So typically, the hippocampus oscillates around 5 to 12 hertz, with an average around 7.5 to 8.5 hertz. What we found, though, is that in the STZ-treated animals, regardless of fluorescein or not treatment, that speed was actually increased up to an average around 10, which could impair the way the hippocampus and other brain areas interact via these rhythmic oscillations. We also found that this increase in frequency that the hippocampus is oscillating impaired the hippocampus's ability to interact with the frontal cortex. So we looked at a measure referred to as coherence, which is a correlation of power changes or oscillatory changes from one brain area to another. So if the hippocampus had a high-powered 8 hertz oscillation, we would expect the frontal cortex in this case to also have that high-powered 8 hertz oscillation. Again, the rating is between 0 and 1, so very much like a correlation. 1 being max coherence or the same signal, 0 being no coherence at all. What we found is that the STZ group, again, was lower than our control animals, despite fluorescein treatment or not. So our results suggest that cognitive impairment can occur due to a deviation from normal glucose. It's not just elevated or lower. It also shows that the hyperglycemia alters the way the hippocampus communicates with other brain areas and impairs its ability to interact with the frontal cortex or send information to the frontal cortex. We plan on doing follow-up experiments where we actually look at the neurons within these brain areas. We have the data, we just haven't been able to get to it yet. And we're hoping that the end of this study, we will have a better idea of how fluorescein, STZ, hyperglycemia, and neuroinflammation all lead to some kind of cognitive impairment. And finally, I just want to do a special thank you to my advisor, Dr. James Hyman, and my lab mates, Emmanuel and Ava, also the UNLV Cellular and Molecular Brain Research Laboratory. Our collaborators did a lot of this work with us. They actually developed the hyperglycemia or diabetic model in rodents. Also UNLV and the GPSA for supporting me and some of this research. And if anyone has any questions or comments regarding this project, I'd love to hear from you. My email is right up there. So thank you, and have a good rest of your day.